Hey everybody, welcome back. Well, welcome for the first time to Shepherd High School Honors Biology. We're going to attempt to cover some material digitally here, so bear with me. Today I want to review some things that I'm hoping you've been working on during your time at home with e-learning, and then get into some new stuff a little bit later on. So let me recap what we have worked on so far. We began our unit here by watching a video about an albino family living in Brazil. You came up with some questions about what it means to be albino, what causes it, how is it inherited. We spent the first part of our semester here going over how it's inherited. We discovered that albinism is caused by a gene, particularly a mutation, and that it's a recessive trait. So you have to inherit 2 copies of the albinism gene in order to be albino, which explains why it can be present in some individuals who are siblings and some who are not. So we worked on some Punnett squares, we worked on some pedigree things, and really came to the conclusion that it's an inherited trait. We then went into what albinism is technically and started talking about melanin. And melanin is, it's a pigment. So back when we were doing plants and photosynthesis, we talked about chlorophyll as a pigment. And all pigments really do is they either absorb color or reflect color. So melanin is the pigment we find in our skin, in our hair, and in our eyes. And there are different genes that control where the melanin is made and how much is made. We watched a little video clip about melanocytes. And melanocytes are a special type of cell that actually produce melanin. So not all cells in your skin produce the melanin. The melanin does get distributed, though, to different layers of the skin and different parts of the body. So what we're going to talk about next is the genetics of melanin on the DNA level, at the molecular level, and how it is that we can make this melanin protein, because it is a protein. And then eventually we'll get into how it is that it doesn't get made. So what exactly is going to cause someone to be albino? Okay, so let's review a few things that we watched in our video the last time we saw each other. We reviewed what melanin was, and then we discussed that there's 2 kinds of melanin found in skin particularly. Eumelanin, which the prefix EU means true or common. So eumelanin is the most prevalent one on Earth. So that includes the brown, the black, and every color in between. Then there's also pheomelanin, which is a special type of melanin that gives red hair, and also fair features as well. So we discussed that not all cells are the same, that there are particular cells called melanocytes that produce the melanin. And by the way, the suffix cyte, C-Y-T, always refers to a type of cell. What is the purpose of melanin? Why do we even have a color to our skin? Melanin protects our cells, particularly from ultraviolet radiation from the sun, ultraviolet light. We're going to talk a little bit later about what happens to cells when they are bombarded with a whole bunch of UV light. This connects a little bit to what we watched in the video, and you guys came up with some questions about why does someone who's albino have a high incidence of skin cancer. So this kind of will begin to explain why. And then also your amount of melanin can change over time. For example, when someone says they have tan skin or their skin is tanning, what's actually going on there is that by exposing yourself to more ultraviolet light from the sun than they normally do, it's a signal that tells your cells to start producing more melanin than usual. The melanin itself will form a cap over your nucleus, so we don't have the right picture here, but I will find the right picture for you, and that cap then protects you from the sun. There is a limit though, like there's probably only so much melanin a person can actually produce. Okay, so getting into next a little bit about DNA. You did a little reading on your own about deoxyribonucleic acid. That is the chemical found in every nucleus of every cell. Its job is to store up all your genetic information and then also transmit it to other cells and then even to future generations. So we're going to talk about how cells make copies of each other in our next unit. The purpose of DNA is to tell a cell what protein to make. So basically DNA is a code, And that code tells the cell what proteins to make. Certain cells will make certain proteins. So for example, if there's gene, let's say, a gene for melanin somewhere in my DNA, and every cell has the same copy of DNA, not every cell is going to be making melanin. My brain, for example, does not make melanin, but my skin does. So different cells will use different proteins, but they all have the instructions on how to make everything. You've heard DNA referred to as a double helix, which means it's two helices wound around each other. So I cannot draw, and there are much better drawings coming up, but you've got a helix and then you've got another helix here. We're going to talk about the structure of that helix. The helix can come apart really easily. We call that unzipping, and that makes a lot of the other cell things we're going to talk about this year possible. So for example, mitosis, which is how cells make copies of each other. Well, in order to make a copy, you have to also copy the DNA too. So the nice double helix shape allows for that to happen quite easily. Okay, so back into the structure, you did a little bit of coloring of DNA. I don't even know how long ago that was. But DNA is a really long repeating molecule. It has a bunch of smaller pieces that we call nucleotides. So that's a really important vocabulary word, a nucleotide. In the picture you see here, this is a nucleotide. Nucleotides will repeat over and over and over again in the DNA structure. There's actually 4 different types of nucleotides, but they have the same basic setup. So a nucleotide has 3 parts to it. The first part is the sugar, and you can kind of see the sugar labeled right here. The sugar has a name in chemistry based on the arrangement of all of its atoms. This is called deoxyribose. Molecules in biology and in chemistry that end in OSE tend to always be sugars. Now this deoxyribose, you notice, has a very geometric shape. It forms a pentagon here with a 5-sided molecule. The corners of that molecule are carbon atoms, but they just don't draw them in because it gets kind of messy, but that's what that means in chemistry. So there's a sugar. It's going to be attached to what's called a phosphate. And a phosphate is a phosphorus molecule surrounded by oxygen. Notice that a couple of the oxygen have what's called a negative charge on them. If you've ever wondered why DNA is a twisted molecule, why the helix forms, while it's like a spiral staircase, and that's because when you put charges that are the same kind of charge, like negative charges, really close to each other, they repel. You've probably heard that opposites attract, and that's true. Pluses and minuses would attract, but 2 negatives together would repel. So what causes the twisting is those negative charges are trying to push away from each other, and it just causes the molecule to spread out. And then the other most important part, one could argue, is the nitrogen base. Sometimes it's called the nitrogenous base, sometimes just base. Base is fine with me. This is your base molecule over here. This is actually only one example. This is called adenine. The adenine is going to be really important when we talk about genes and when we talk about codes. So altogether, there are 5 different atoms that compose DNA: carbon, hydrogen, oxygen. And if you remember back from your Africa unit, those are the same basic components of a carbohydrate. We also add in here nitrogen, which is a component of a protein. And then we also add in the phosphorus from the phosphate group, hence the name. So those 5 atoms are the only types of atoms and elements that you'll find in DNA, but the way they're structured is what gives you a unique molecule. Okay, a few more pictures here reviewing the structure of DNA. This is a good cartoon version where they've left off a lot of the details, but you can clearly see the ribbon-like structure as it goes around. So you've got one ribbon surrounded by another ribbon, twisting. Now the ribbon is actually called the backbone. So the backbone of DNA is pretty boring. It's just the sugar, deoxyribose, and the phosphate repeating after each other. So I will write sugar for S here, so it'll go sugar, phosphate on one side. It's going to go sugar, phosphate on the other side, and it just happens over and over again. This molecule is really long. I would say that it probably happens billions, maybe 3 billion times over and over again in one cell. So obviously these molecules are really small. The important part, though, is the inside, which they've highlighted with letters A, T, G, and C. These represent the 4 examples of the 4 types of bases found in DNA. A stands for adenine, T stands for thymine, G for guanine, and C for cytosine. They all have that same "ene" ending, and that has to do with the nitrogen. So in chemistry, we name things according to things they have in common. So these all have something in common, hence the same suffix. But we usually just call them A, T, G, and C for short. Okay. Next, the two halves of the double helix are bonded together. And they're connected together by an attraction between molecules called a hydrogen bond. But the bonds that hold the two sides of the double helix are kind of weak on their own, and that kind of allows it to open up and break apart really easily, which becomes important when we want to make a protein like melanin. So how do we know how the two sides are connected? Well, they're connected based on what's called complementary base pairing rules. And it says that the way they're structured with all their available electrons, A and T tend to bond together because they have complementary pairs of electrons, and G and C bond together. So for every A, there would be a T, and for every G, there would be a C. So that's how, in the picture from the previous side here, the pattern that you see, I know I wrote all over it, but you can kind of make sense and see that G and C are bonded together and A and T are bonded together, and that happens over and over and over again. Okay, so one of the skills that we'd like you to have is, if I give you one side of the double helix, could you tell me what the complementary half is? So if this is ATG at the top, the complements would be what pairs with what. So A pairs with T, T pairs with A, G pairs with C, A pairs with T. Next we have another G, a G, A, and C. So all you have to do is find what's the complementary other half of the double helix. The other skill is, if you know approximately how much of one particular type of base there is in the nucleus, could you then calculate what the other bases are? So for example, if there's 20% adenine, on the previous slide we said because A and T bond together, for every A you will have a T. So if there's 20% adenine, there should also be 20% thymine. If you add up 20 and 20, that gets you 40%. 100 minus your 40% ends up with 60. If you split 60 equally between G and C, you're going to get 30 and 30. And then those four percentages have to add up to 100, otherwise you've done something wrong. In real life, though, they're not exactly equal because there's a little thing we call mutations, where sometimes bases don't bond as they should. Okay, included a few more artists' interpretations of what DNA looks like. You've got your ribbon model on the left, your computer-generated model. This one's really accurate, shows you exactly how the atoms connect together. You can see there's a lot going on. And then the chemical formula, or the chemical structure in the middle here, kind of partially done for you. But I just want you to notice the backbone. So notice we have our phosphate in yellow and our sugar in blue, and it just repeats over and over and over again. The other side is the same, except you might notice that the other side is upside down. You can really see it when you look at the sugars. On the left-hand side, we see the oxygen at the top. On the right-hand side, the oxygen is at the bottom. And that's true in real life. In actuality, the two sides are called anti-parallel, if I can write that out. Probably not. Anti-parallel, which means they are parallel lines, but one of them's upside down. And that has to do with the way the bases connect. When T and A want to bond together, A basically has to flip upside down. These little red dots that you see in the middle, these are the hydrogen bonds. Notice there's two rows of them, so T and A will share two hydrogen bonds, and G and C will actually pair 3 hydrogen bonds together. That's not really that important for you to know. Just know that that's where the attraction is. Okay, so to wrap it up here, a little review of DNA. DNA is a sequence of genetic information that forms up all your genes. In a human, we have probably about 25,000 to 30,000 genes, grouping in all those little nitrogen bases that you find. The gene is a code, and that code is going to tell us what protein to make. The proteins we make, like melanin, become your phenotypes. Remember, from genetics, your phenotype is your physical appearance of a trait. It's how it looks. So like we would say, brown hair, that would be a phenotype. And then what we're going to talk about in the next little part here is that proteins are made not inside the nucleus, but they're made in a little organelle called a ribosome. So in our next part, we're going to talk about how does the DNA actually become the protein, or how does it become the melanin. Okay,